All right, so this is the urinary system, chapter 26. We're in the beginning of the fourth unit here. And we just have urinary and reproductive in terms of organ systems, but then we'll also do development to sort of wrap things up. And so, urinary system, I know a lot of this is done in lab, or will do in lab, but as typically the case, it's good to have go over the anatomy a little bit to understand the physiology of how it works. And so basically, the primary organs or structures involved in the urinary system are the kidneys, of course. The kidneys are the location where urine is manufactured. And urine is primarily produced to help with waste, removing of waste products, maintaining blood pH, blood pressure, and things like that. So we'll talk all about urine production. That'll be the primary physiological thing we'll look at in this in lecture. Ureters are then the components that are attached to the kidneys that are basically a transportation device to get it down to the urinary bladder, which is a temporary storage device, and then the urethra is the last portion that allows for excretion or release of the urine. So those are the primary structures, not as nearly obvious as many structures as we saw, like with digestive, for instance. But again, the three functions primarily of the urinary system: excretion, obviously, is a big one, removal of organic waste products from the body. So elimination also, and discharge of waste products. But then homeostasis, homeostatic regulation of blood plasma and solutes, so making sure your ion concentrations are good, all your plasma is good. And so you get a lot of blood flow going to the kidneys, and the kidneys are basically producing a filtrate, which is urine, which can contain waste products and water, but also help to maintain homeostasis within your blood. So that's really important, obviously. So those are our functions. Kidneys, obviously, as I said, are the organs that actually produce and manufacture the urine. The urinary tract then is the ureters, bladder, and urethra, which is the pathway for release, essentially, of the urine. And then urination is the process of urinating the urine, which is also micturition, or the micturition reflex is what we'll look at, which is as the urinary bladder fills, it's going to contract because it's smooth muscle. Transitional epithelium is smooth muscle. It's going to contract and push urine through the urethra and then out of the body. And we'll look at that process as well. So the homeostatic functions, as I said, primarily this is a big function of the urinary system. Number one, regulating blood volume and blood pressure. And they say, well, how can the urinary system maintain blood volume and blood pressure? Well, a lot of it has to do with water, first of all. Water is a big component of the urine, so there's more or less water. Water is a big component of blood, as you remember, so you're going to impact blood volume as well as blood pressure, right? The more fluid you have in your system, the higher the pressure, less fluid, lower the pressure. So the kidneys actually have the ability to adjust the amount of water being lost in the urine to make sure that blood volume and blood pressure are where they need to be. Also, EPO, erythropoietin and renin, are hormones essentially that are released. Erythropoietin, for instance, stimulates red blood cell formation. Again, that's going to impact blood volume, right? More red blood cells, more blood volume they're going to have, and that's going to impact the blood and blood pressure. So those are really important in terms of maintaining homeostasis of those components. Also regulating plasma ion concentration, sodium, potassium, chloride, calcium. All these things can be maintained in balance. If you have too much sodium, or too much potassium, or too much chloride, you can lose these things in the urine. If you have less of these things, you can lose less in the urine and hold on to more. We talked about calcium ion levels. Way back in AMP1, we were looking at the bone and also in the endocrine system and how calcitrol helps with maintaining calcium ion levels, and you can either release more through the urine or less depending on the concentration of calcium ions. Also talked about like blood sugar, right? It's one of the things we said about diabetics, for instance, right? They have high quantities of sugar in their blood, obviously, right? Because their insulin is not doing its job, or it's not being released at all, and blood sugar is going high and remaining high. So what's the body going to try to do? Well, it's going to try to get rid of that excess sugar if possible, since it's not going into the muscle, not going into the liver. How's it going to get rid of it? Well, one way is to try to excrete it in the urine. It's try to lose more in the urine. So we said that was one of the hallmarks of someone with diabetics is extreme urination and extreme thirst, because their body's producing a lot of urine to try to get rid of this excess sugar, and therefore when you lose a lot of urine, of course you're going to stimulate thirst, and so you drink more. Urinate more, drink more, urinate more, as you're trying to lose all this excess sugar and maintain homeostasis. So anyway, so it can regulate plasma ion concentrations by controlling what's lost at the urine. So other things help stabilize blood pH. Again, this ionic composition of hydrogen and bicarbonate ions can help impact blood pH, and blood pH needs to maintain a certain level, as we talked about with the blood, way back when we looked at the blood in the first chapter of the semester. Conserve nutrients, right? So it can hold on to things that are very useful, and also assist the liver in sort of filtering and detoxifying, getting rid of excess potentially toxic components from our blood. So here's just another shot of the anterior view of the trunk, where you can see the kidneys, you can see the adrenal glands right on top of the kidneys. We talked about those back with the endocrine system. And so what we want to do first, and again, a lot of this is run over in lab, so I'll just go through these pretty quickly. We want to take sort of a sectional anatomy of the kidney so we can look at the different structures. And so the first thing you can see is the renal sinus. Then within that, above that, you have the renal cortex and the renal pyramids, probably easiest to see in this location. So the sinus you can see right here in this location. And then you can see the cortex is this section, and that's where a lot of urine is going to be manufactured, essentially, and what are called nephrons. And then the medulla, or pyramids, are essentially in that location, as you can see there. Ultimately, the urine that's being manufactured is going to drain into the major and minor calyces, which are in this area. That's going to then drain into the renal pelvis, and then ultimately down the ureters, where it will be sent to the bladder, where it will be held until it's excreted from the body. All right, so those are a lot of the structures you can see. This, again, the renal lobe is the pyramid, the cortex, and this is where urine is manufactured. And I talked about the pelvis, where the funnel-shaped chamber where it fills and connected to the urinary, which is ultimately draining the kidney, the urine that's being manufactured. And so there is an actual frontal section of the left kidney. You can see a lot of those structures as well. All right, so since the urinary system is essentially about filtering the blood, obviously there's the most significant blood supply going to the kidneys. And so the kidneys receive about 25% roughly of total cardiac output. So 1,200 mLs of blood flows through the kidney each minute. So you're seeing a lot of blood pumping through these kidneys. As the blood passes through these kidneys, you're going to see this filtration process that occurs at the nephrons and the formation of filtrate, which ultimately will become urine. And so the kidney receives blood through the renal artery, of course, and that's how this process begins. And once the renal artery enters the kidneys, it starts branching into smaller and smaller components. Again, this is sort of a lab item, but segmental, then renal, interlobular, obturate, and then ultimately down to the small structures. So here you can see the renal arteries, so blood is initially being centered here, and then again, as you can see, it's just going to keep channeling into these smaller and smaller branches until it finally is going to reach up here at the cortical radiate artery, all right? And that's where ultimately, at the end of that, we're going to see urine manufactured. So you can see it keeps branching, getting smaller and smaller. Now, the cortical radiate artery branch into what's called an afferent arteriole, and this is the last segment that delivers blood into the nephron. The nephron, as I said, this is ultimately where urine is going to be manufactured, and then the cortical radiate veins also take blood the other direction and empty back out into the renal vein. And so here you can see a nice image of that. You can see the arteries turning into the caudal radiate arteries, and then those are going into the nephron with the glomerulus, which are these little balls. That's a very good circle, but those balls right there is where they're located. Okay? And so this shows, again, you can see the renal cortex up here, right, and then the pyramid down here, and you can see these, we'll talk about these in a little bit, but these purple guys are basically sort of the drainage ducts where the rest of the processing and reabsorption of water is occurring, and then ultimately it's going to drain down into the minor calyx, and that's where the urine will fill and then be sent down to the ureters and so forth and so on in terms of urine production. So this happens at all these locations, so you can see there's a lot of locations for urine production to occur. And this is a great slide here because it shows the pathway of blood. Starts at the renal artery, ends at the renal vein. You can see it just keeps branching smaller and smaller to segmental, interlobar, arcuate, cortical radiate, afferent, into the glomerulus, out the efferent, and then back out the veins. Right, as the blood passes through this nephron is where the filtrate is being produced, and the blood returns back to the venous system. Renal nerves, of course, innervate the kidneys, so there are nerve endings located in this location as well to allow for, obviously, the autonomic nervous system. Sympathetic innervation is going to adjust the rate of urine formation, right? And it does this by just changing how much blood flow can enter the nephron, right? And so if you alter the pressure, you're going to alter the amount of blood flow. If you get more blood, obviously you're going to produce more filtrate, less blood, you're going to produce less filtrate. And so that's going to impact what's happening. Also stimulates the release of renin. We'll look at the renin-angiotensin system a little bit later, which helps with restriction of water and salt and things like that by stimulating reabsorption, essentially, in the nephron. So we'll look at those processes as well. Okay? So nephrons, then again, are these microscopic structures where the urine production essentially begins, right? It's at the very beginning of this process. And it has a renal tubule, a renal corpuscle, contains what's called a glomerulus, a capsular space, and you can see all of these structures in the nephron. And so here you can see the afferent arterial blood starts from the arterial system entering this way, and you can see the green arrow, which indicates filtrate is being produced. And then here it goes through all these structures, and then you can see the efferent arterioles where it then moves back out of the glomerulus. So you can see those structures there, and then as the material moves, you can see the filtrate is going to pass through this system. It's called a convoluted tubule. You have a distal convoluted tubule, then a proximal convoluted tubule. You also have this thing called a nephron loop, and we'll look at the different components that occur at these sections. So, nephron then is this renal corpuscle. It has this glomerular capsule or Bowman's capsule, which is a chamber where you're, basically made of capillaries where you can see filtration occurring. And so the glomerulus is these 50 intertwining capillaries and ultimately drains from an afferent arteriole into this section, leaves via efferent arteriole, or it returns to the venous system. But this glomerular structure is really important because these glomerular capillaries are fenestrated. Fenestrations, right, means openings, and it contains pores, right, or openings that allow for the formation of the filtrate. The best example, and we'll talk about this in a minute when we look at it, but when you think about how these glomerular capillaries, these fenestrated capillaries work, is to think of like a coffee filter, right? You have all the things on the one side of the filter, you pour water over it, and on the other side is the filtrate. And that's sort of what these fenestrated capillaries are like. They're going to allow water and smaller things to pass through, but the larger things are going to remain in the capillary bed. So filtration, again, occurs in this renal corpuscle, and it's driven by blood pressure, right? The amount of pressure is going to force water and dissolve solutes out of the capillaries, out of the glomerular capillaries, into the capsular space, right? And this produces this protein-free solution. As the proteins are remaining back, again, think of the coffee filter example, and the filtrate is being produced. kind of like a blood plasma, right? Mostly water, but has these smaller dissolved solutes and things like that in it as well. And so again, like coffee being produced from a coffee filter. Okay? And so this is a great image where you can see that happening. And the red arrow down here at the bottom is showing where it begins, right? The afferent arteriole. You can see smooth muscle around that afferent arteriole. The reason that is so important is because it can constrict, it relax, vasoconstriction, vasodilation, which can either increase or decrease the amount of blood flow that's moving through this component. And as it passes through here, you can see all these fenestrations along this endothelial lining, and then basically, depending on the amount of pressure, that's going to produce filtrate that's going to go into this capsular space, which is this, right? And so all this filtrate's going to go into this capsular space, which is here, right? All this in here, and as it fills, it will then move out the proximal convoluted tubule. That's sort of the first step in this filtration process, going through that structure. So that's a really great image. You can also see the DCT, or distal convoluted tubule, over on this side. So the nephron then again also has this renal tubule. After the initial structure where the filtrate is formed, you have the proximal, distal, and then nephron loop, PCT, DCT, and loop of Henle, which are really important in the ultimate formation of urine, because initially we just call it filtrate. Then it has to pass through this renal tubule where a lot of water is reabsorbed, other potential ions that can be used are reabsorbed, and then ultimately it forms urine. So again, you have proximal convoluted tubule and a distal convoluted tubule, both located in the cortex, and the loop of Henle can extend partially in medullary cavity, as we'll see in a minute. Okay? Let's see if I can get that image real quick. Yeah, here's a pretty good shot of it, right? Here you can see the proximal convoluted tubule over here, right? Distal one over here, and then the loop of Henle right there. Okay? All right, so what are these things all about? What are these renal tubule components all about? Well, first you have the proximal convoluted tubule, and again, this is the first segment that initially takes the filtrate from the glomerulus. So basically the point of connection with the afferent arterioles with the glomerulus, simple cuboidal lining, and functions primarily in reabsorption. We're going to see in a little bit, the amount of water used to initially produce the filtrate is very large, and on the order of gallons, and you need to reabsorb a lot of that water. Okay, and so this lining has the ability to reabsorb that water and hold on to it instead of losing it completely. The distal convoluted tubule, right, it's after the loop of Henle, is the third segment, has a smaller diameter than the PCT, and lacks microvilli. And so what we see at the DCT are more, you see some secretion of ions and toxic components, right? These are going to help the liver with a detoxification process. We see reabsorption of certain sodium and calcium ions if the body wants to hold on to those. And then again, reabsorption of water. That's a big process in these tubular pathways, is reabsorption of water. So the loop of Henle again has a descending and ascending limb, one thicker and one thicker. So this is a nice cross section through each of the components. You can see the renal corpuscle really well here. You can see the glomerular capillaries, and you can see the capsular space really nicely well that has been produced. And then you have the PCT, the nephron loop, and the DCT. So you look at a nice cross-section of what they look like, the different sizes of the cells, and then characteristics, length, diameter, and function, right? And so that's really important. The corpuscle obviously is producing the filtrate, and then what's happening the rest of the way has a lot to do with reabsorption, right? Reabsorption and reabsorption. So make sure you read through this and have a good understanding of what's happening with each of these segments. I think it's going to help you understand the overall picture. And so here's a nice shot again of that anatomy of these different components of this nephron and how urine and filtrate ultimately is absorbed or produced. So three functions of renal tubule, again, reabsorb useful nutrients that happen to enter the filtrate. So if there are things the body can hold on to and use, it will, and reabsorb them in the renal tubule. Reabsorb more than 90% of the water. So you're only using about 10% of the water that's initially used to produce the filtrate. And then also secretion of waste products that maybe didn't enter through that process previously at the glomerulus. All right, so each nephron then empties into what's called a collecting system. This collecting system or ducts receives these fluids and ultimately is going to drain it into the minor calyx. But this is the last sort of stage. And again, maybe you see a little water reabsorption or solute reabsorption, but ultimately the last thing is to deliver the urine to the minor calyx, where it will then ultimately drain into the ureters. All right. So this ultimately, this collecting system is determining the final concentration and volume of the urine. So this is an important component in the overall system. Right? And there you can see the collecting duct and the papillary duct, the last portions of this system. All right. So now that we understand generally how a nephron is organized and the different components and how urine is formed for the most part, we want to look at some different types of nephrons. There are actually two types. The vast majority of them are called cortical nephrons, 85% of them, and most of them are located, these are located mostly in the superficial cortex of the kidney. The big difference between the cortical and the juxtamedullary is that the nephron loop is pretty short in the cortical nephron. In the juxtamedullary nephrons are much longer, and probably is mostly just an efficiency of space type of scenario, allowing you to have different components. But you can see the cortex up here, you can see the medullary cavity down there, and you can see the two different types, cortical and juxtamedullary. And you can see how much longer the loop of Henle and the juxtamedullary component is. But ultimately, they're both draining, producing, and draining urine into the minor calyx, where it's initially going to be sent to for drainage of the kidney. All right. So those are just images you can look at, looking at the blood flow and the pathways. All right, so next thing I want to do is look at renal physiology, a nice understanding of the structures and basically how urine is produced. And so what we want to do then now is look at renal physiology. And if you think about it, the overall goal of urine production is to contribute and maintain homeostasis in the blood. Okay? And so there are several things that are involved in that process. Number one is regulating the volume and the composition of the blood. And so in doing that, it's going to excrete 3 metabolic waste products: urea, creatinine, and uric acid. These are ultimately eliminated in the urine, and of course, part of that is water loss. That's sort of the vehicle for release of these components is the water in the urine. So that's how these things are lost, essentially. Urea is a very abundant organic waste product as a result of the breakdown of amino acids, and of course amino acids are being broken down quite a bit for the body, and as it uses, it's producing about 21 grams of urea each day. Creatinine, as you recall, we talked about creatine phosphate, and AMP1 was related to the muscle contraction, so obviously producing a lot of that. And then uric acid is also being formed in recycling of RNA. So you have a lot of components that there's no way for them to get out, and you need to pass through urine from that standpoint. So those are some essential components. Some other things, of course, we have filtration, reabsorption, secretion that we want to talk about as well. And so you can see this shows like overview of the process. Starts here, right? And then as you can see, all passes all the way through and ultimately ends down in this location. And so that's your pathway of it. It's like primarily just medullary nephron. All right. So basic processes of urine formation then, as I said, filtration. reabsorption, and secretion. That's what needs to happen along the way. Filtration is essentially, again, the process of producing the filtrate. And you have pressure that forces water through the pores on the membrane of the glomerulus. The small solute molecules that pass through the pores, everything else is retained behind those walls. And that's essentially how filtrate is being produced. Certain areas like the liver, for instance, you have larger pores that can allow for larger proteins to enter, but here we're seeing a specialized membrane that's restricting those proteins from being filtrated. Okay? Reabsorption and secretion at the kidneys, diffusion, osmosis, all these things are happening to allow for reabsorption and secretion to occur. And that's ultimately the processes that we see. And so if you look specifically at glomerular filtration, we're going to see how this works and how it actually is passing across this filtration membrane. And so to understand that, when you look at the anatomy of this glomerular filtration membrane, and you have a capillary component, of course, you have a dense layer, and then you have these slits on the membrane itself. And so here you can look closely at a section, obviously, and you can see this is inside where all the blood is coming, and this is in the capsular space. And so the blood needs, or the filtrate needs to pass through these locations into that capsular space. And so that's what we want to look at, see how that happens. Okay, so again, glomerular capillaries are fenestrated capillaries, meaning they have pores in them or openings, fairly small, 60 to 100 nanometers in diameter. Obviously, red blood cells are not going to be passed through there, but solutes and plasma proteins will sort of work its way through those fenestrations. The next layer, if you look at this image again, you have what's called a dense layer after that, which is here in the middle. Okay, the dense layer is a little more selective, right? Only smaller plasma proteins, nutrients, and ions are going to pass through here. And then finally, the filtration slits have gaps that are 6 to 9 nanometers wide. And that is going to prevent passage of most small plasma proteins. So this is defined as the filtration layering. And you can see that's basically, sort of think of it like as a 3-tiered filtration, coffee filter or something like that helps you understand how that works. And this filtration by the glomerulus, again, is governed by or driven by pressure, right? The pressure that's arriving at the glomerulus and the pressure that's inside the capsular space, right? Those both things are going to drive this pressure and how much filtrate is ultimately used. So you have the hydrostatic pressure, which is in the fluid, and then you have just the pressure of the materials in the solution itself. These things are all going to contribute to that. And so here you can see exactly what I'm talking about. This is inside the glomerulus. You can see the larger plasma proteins staying back and solute being produced. The pressure in here is what we call glomerular hydrostatic pressure. So the pressure inside that is about 50, right? It's about 50 millimeters of mercury passing in to the capsular space. But you also have to consider that there is pressure of the blood that's going to be driving it, and then capsular pressure that's also going to be driving the other direction. And so ultimately your net filtration is only 10, even though 50 is what's driving it up, because you have these other components working against the pressure gradient, so to speak. So ultimately you only have about 10 millimeters of mercury in terms of net filtration pressure producing filtrate into the capsular space, and then ultimately going into the proximal convoluted tubule. So that's what net filtration pressure is, right? The pressure forcing water and dissolved materials out of the capillary into the capsular space. Okay? And again, this is the difference between the pressure, the hydrostatic pressure and the blood's pressure across those glomerular capillaries. Okay? And so GFR, glomerular filtration rate, is the amount of filtrate the kidneys produce each minute. As I said before, it's about 10% of fluid delivered to the kidneys enters the capsular space. Okay? And so the glomeruli are generating about 48 gallons of filtrate a day. So can you imagine if you're producing 48 gallons of pee, right? I mean, that's not going to be good. That's why reabsorption is so important. And 99% roughly is reabsorbed in that renal tubular pathway and held onto instead of being excreted, right? So filtration pressure then, as I said, is what drives this whole thing. Right? That's the first step in kidney function. You have to have the appropriate amount of filtration pressure at the glomerulus in order to produce urine. And so if filtration does not take place, then waste products are not going to be excreted. You're not going to be able to maintain your blood pH. And so this whole thing is driven by pressure. And so GFR, which is how much filtrate you're producing, depends on the pressure, right? And there are different ways to alter the pressure and therefore alter GFR. Okay? So how can you control GFR, glomerular filtration rate? Autoregulation, specifically at the local level, hormones can impact it, and then autonomic sympathetic division. So you have what's happening specifically at the glomerulus, you have hormones, and you have the sympathetic division of the autonomic nervous system. 3 different ways of controlling GFR. Okay? And so first thing we can look at is autoregulation, local level, right? And this is important because this is saying that you can maintain GFR despite changes in blood pressure and blood flow. As you've known, we've talked about before, your blood pressure is not always 120/80, right? It's going to change depending on if you're lying down or if you're up moving around or if you're sitting, depending on the different scenarios, your blood pressure is going to change. Does that mean you want to necessarily change the blood pressure that's at the glomerulus? Not necessarily. You want to be a little more consistent there, okay? And so you can maintain GFR despite whatever changes in blood pressure are occurring systemically outside of the body. And to do that by changing the diameter of the afferent arterioles, the efferent arterioles, and the glomerular capillaries, right? You can change that and adjust depending on what's going on outside the body. So GFR remains constant when systemic blood pressure rises, right? So stretching that can occur, constriction arterioles can also decrease blood flow to counteract that increase in blood pressure and change in blood flow. Okay, so that's a really important one. And so here you can see autoregulation, right? Homeostasis is normal GFR. Let's say GFR starts to go down, right? Because maybe your blood pressure is going down, and so decreased GFR is going to result in decreased filtration, decreased urine production. If you're losing that, obviously you're losing the ability to, keep your body in homeostasis. So, in response to that, dilate those afferent arterioles, right? Constrict the efferent arterioles, and you're going to get more blood into that glomerulus, increase glomerular pressure, and you're going to return back to normal GFR. Okay? So that's a good example of how you can, autoregulation of maintaining GFR. Okay. What about hormones? Hormones can also regulate glomerular filtration rate. Primarily, what I want to look at is the renin-angiotensin system. Renin-angiotensin system. When you have a drop in GFR, so you're not producing as much filtration, right? Glomerular filtration rate, GFR is dropping. That causes the structural glomerular complex to release renin. Renin has several steps. That's why they call it the renin-angiotensin system, because renin converts inactive angiotensin to angiotensin 1. Angiotensin 1 is then converted into angiotensin 2 by ACE, or angiotensin converting enzyme. And then angiotensin 2 can act at the nephron, the adrenals, and other locations in CNS to stimulate pressure. And once it's active, you're going to see constriction of the efferent arterioles. That's going to elevate glomerular pressure, stimulates reabsorption of water and sodium ions, trigger release of ADH, right? Antidiuretic hormones also going to stimulate reabsorption. So all this stuff is trying to help increase pressure, which is going to help then get GFR back up. So renin-angiotensin system is working that way. Thirst, right? Simulating thirst, getting more water into your system, that's going to help increase pressure. increased sympathetic motor tone, right? Increased cardiac output. There you're going to see an increase in pressure as a result of that. So all these things are elevating arterial pressures throughout the body. That's going to help you get GFR back up, right? It's all about pressure. Pressure drives this thing. So if you get pressure back up, all these things the renin-angiotensin system do, you're going to get GFR to get back up, right? So that's another hormonal system that allows this process to work. And so this is a nice slide that's sort of showing when you have a reduction in GFR, how the renin-angiotensin system is basically trying to drive pressure back up. If you get pressure back up, you're going to get pressure in the glomerulus back up, and that's going to increase GFR. Okay, and so this shows that process of the renin-angiotensin system. All right, so make sure you look through that. Autonomic regulation, again, is pretty similar. This is more directly related to sympathetic activation. It can constrict the afferent arterioles, decrease GFR, and slow filtrate production. So this is a way of, if you want to slow GFR, you can do that through autonomic regulation via the sympathetic division. All right, so now we're moving on to reabsorption and secretion. Again, reabsorption, purpose of it is to recover useful materials from the filtrate. Secretion ejects waste products and toxins, and ultimately sort of the last steps before urine formation occurs. And then urine production, ultimately, in a healthy adult, is producing about 1,200 ml a day, which is only 0.6 of the filtrate that was initially produced. So again, that reabsorption of water is essential in that process. Okay? So diuresis then is the elimination of urine. Diuretics are drugs that may promote water loss in the urine, right? So diuretic therapy can reduce blood volume, blood pressure, right? And help, these are all going to help with increasing loss of water. Composition of normal urine, again, from the filtration, absorption, and secretion activities that occur at the nephron, are going to work on these processes together and produce these components. Urine depends on, what exact processes are happening in the body, but ultimately we're seeing a clear sterile solution. There is some urobilin pigment in there generated from the kidneys. And urinalysis can be very useful, right? You can look at a urine sample, and there's all kinds of things in your blood, from hormones to, that are ultimately in the urine that you can look at to get an idea of what's happening inside the body. So urinalysis can be very, very useful. And so here are some of the things, general characteristics of normal urine, from pH to volume to color, bacterial content, etc. And then here are some of the things you can, compounds you can see in the standard urinalysis. Obviously, urea, creatinine, ammonia, uric acid, things we've talked about that you'll find in the urine, and you can see the significance of some of these things. So this is a nice chart to see what types of things you're seeing in the urine. All right? Sodium, potassium, calcium, etc. All right, so this next slide, I'm going to leave on here, but I'm not going to go over this. This is just sort of summary of the 7 steps of renal function. So you want to make sure, again, that you can sort of go through all of these steps and have a good understanding. Again, this is kind of a summary. We've really sort of been over this, but make sure you have these 7 steps in terms of the flow of urine from its original formation to reabsorption, to the collecting ducts, and then on out into the last component, and then urine production. And you can see all those slides in the summary of urine renal function there. All right, so on to the sort of home stretch here in the last 20 or 30 slides here is going to have to do with urine transport, storage, and elimination. So what are some of the things we're going to see here? Well, obviously, this is after the kidney, so we're talking ureters, urinary bladder, and urethra. Right? And basically, these are the way that urine is transported, stored, and then ultimately eliminated. You can see the ureters in this image, urinary bladder. I didn't really show the urethra in this image. But first is the ureters. The ureters, again, are a pair of muscular tubes. You can kind of think of the esophagus, right, as a muscular tube connecting your mouth to the stomach. It's kind of like a muscular tube extending from the kidneys down to the urinary bladder. The retroperitoneal, of course, this is the kidneys are. And they pass through the bladder wall at a bleeding angle and basically cuts to the backflow of urine when the bladder starts contracting and trying to push urine out of the body. The ureters, again, have an inner mucosa, which, of course, is transitional epithelium, just like we're going to see at the urinary bladder. You have a smooth muscle layer to help push and move the urine down. And then, of course, you have an outer connective tissue layer. And so there's a nice shot, transverse section through the ureter. You can see the smooth muscle, fairly thick. You can see the transitional epithelium as well. And it'll look a lot like what we see in the urinary bladder. So peristalsis, we talked about during digestion, begins with the renal pelvis, sweeps along the ureter, and forces urine towards the bladder. This happens every 30 seconds or so, right? And so peristalsis, we saw this similar process we talked about in digestion. Okay, so then once it moves from the ureters, it empties, of course, into the urinary bladder, which is a pretty large, hollow muscular organ, and essentially a temporary storage device, right? The bladder can hold about a liter of urine, and then as it fills, it's going to distend, and the smooth muscle will be stimulated, and then it's going to start contracting, and that's when we'll release it. We'll talk about that in the nicturition reflex in just a second. But you can see where the bladder is located. That's more of an anatomy thing. If you look at it, you can see that in this location, this is a male anatomy of the urinary bladder. You can see the ureter sort of filling into the bladder. The female bladder sits right below the uterus. And also, big difference between male and female is the length of the urethra. Male urethra, because the penis is much longer than the female urethra. And so that's why it's very common for women to have, like, UTI, urinary tract infections, UTI, or those type bladder infections, things like that, because it's a much shorter avenue, and it's just easier for bacteria to work its way up in that look to the bladder. And so here's a nice cross section of the bladder. You can see the smooth muscle, or it's also referred to as the detrusor muscle. You can see the ureter sort of stubbing into it, and then this area is sort of like a triangle called the trigone. Is where it's like a funnel, basically, where the urine is essentially funneled down and will then travel down the urethra and then exit the body, of course. So I talked about the trigone already. It acts as a funnel, so we've been over that. The neck of the urinary bladder contains an internal urethral sphincter, which is basically smooth muscle, which helps control the discharge of urine into the urethra. And again, we have parasympathetic as primarily controlling urinary bladder in terms of innervation. Histology, this again is more of a lab thing, but again has smooth muscle called detrusor muscle. Of course, it has transitional epithelium. And you can see right here, here's the transitional epithelium, the submucosa, and you have this nice large detrusor muscle or smooth muscle that contractions for that component. The urethra, again, extends from the bladder to the exterior of the body, and this is the last component. Talked about the difference between the male and the female. The male extends from the urinary bladder to the tip of the penis, and has what's called a prostatic urethra. It basically passes right through what's called the prostate gland. We'll talk a little bit more about this in reproductive system, but essentially one of the challenges that older men can have is the prostate enlarges. As the prostate enlarges, this urethra gets smaller. It's sort of compressed. And so it can make urination more difficult if a person has an enlarged prostate. And then ultimately, again, that's leading through the external urethral orifice and out the body. And so you can see what I was talking about again. Here's the prostate gland, and you can see the passageway and how it goes basically right through the prostate. And so that can be impacted potentially. All right. So we're moving along. The female urethra, as I said, is very short. It's from the bladder to the vestibule, and again, this is why it's easier for women to get bladder infections, UTI, and things like that because of this short urethra. And you can see that right there. Okay. And we'll talk again more about this anatomy, female anatomy of the reproduction, but you can see it directly below the uterus at that location. And then again, the external urethral sphincter is essentially a valve that's under voluntary control, right? And so you have resting muscle tone, and then if you relax, that's going to produce or allow micturition, which is the excretion of the urine, and allow for that to occur. All right, so I think I've been over these structures, so we're good on that. Here's the transverse sections of the female urethra, as you can see that. Now, one of the last things we want to look at here is the micturition reflex, or urination, right? And basically, it's a stretch reflex. Just like we've seen other types of reflex, as the bladder fills with urine, obviously the bladder is going to be distended, right? You know the transitional epithelium can spread out. The stretch receptors in the urinary bladder then basically send signals into the brain and allow for this contraction to occur. Okay? And so interneuron relays the sensation to the thalamus, projection fibers deliver that sensation, and then you get voluntary relaxation of the external urethral sphincter in that process, right? But it begins with the stretch receptors. Typically around 500 ml volume will start triggering the micturition reflex. But again, remember, typically that's under voluntary control. And so here's a nice overview of this process. You can see, number one, as the distortion begins on the bladder, it's sending an impulse up to the spinal cord, that's sending an impulse back down, and you're going to see constriction of the bladder. As it constricted, it's pushing urine down through, out into the urethra, and then finally you have this controlled skeletal muscle allows for that. Now, as part of that process, right, you are also getting connection going up to the brain, and that's what we can see up here. Projection fibers relaying that information from the thalamus to the sensation and through the cortex. Then you also have an interneuron that can relay that information to the voluntary relaxed external urethral sphincter. So it's a reflex process stimulated by stretch receptor that's ultimately under voluntary control. Now, infants lack voluntary control over urination, as you know, and those corticospinal connections that I just talked about have not been established yet. Right? And some kids, it takes longer than others, or shorter than others, to when they can have that sort of regulation and control. And of course, incontinence then is the ability to control urination, and it just could be developmental, it could be trauma, it could be aging. A lot of things can impact and cause incontinence to occur. And so what are some of the things we see related to changes with age as people age as part of the urinary system? A lot of things happen, pretty typical what you might expect. Fewer nephrons, which is going to result in smaller GFR. Body's not as sensitive to ADH, which is an antidiuretic hormone, or maybe not as much as being released. And then, of course, problems with the micturition reflex, muscle tone loss, which can lead to incontinence. Micturition reflex control can be completely lost, of stroke or other types of problems. And I talked about enlarged prostate in the male and how that can impact things. So anyway, those are some of the things that are associated with impacting the urinary system. So that's it for the urinary system. Next lecture will be reproduction, and then we'll finish up with development.